Today we're going to explore fungi, a group of organisms that often gets overlooked but plays a massive role in how ecosystems function and how humans live. When people hear the word fungi, they usually think of mushrooms, but that's just a small part of their story. Fungi includes yeasts, molds, and many species you've never seen because they spend most of their lives hidden in soil, wood, or living tissues. They've been on Earth for hundreds of millions of years and were some of the first organisms to thrive on land. As we go through this topic, we'll focus on what makes fungi unique, how they get their energy, how they reproduce, and why they are essential in ecosystems, agriculture, medicine, and even human health. Fungi represent an incredible diverse group of organisms that falls under the kingdom fungi, also called Eumycota. Based on fossil evidence, fungi have been on Earth for roughly 450 million years, which means they are already well established when plants first began colonizing land. All fungi are eukaryotic, so their cells contain nuclei and membrane bound organelles, and they have heterotrophic, meaning they must obtain nutrients from their environment rather than making their own food. Fungi have flexible reproductive strategies. Some reproduce only asexually, while others alternate between sexual and asexual reproduction. Most fungi produce enormous number of haploid spores, which helps them disperse widely. Ecologically, fungi are essential because they act as decomposers, breaking down organic material and recycle nutrients back to the ecosystem. Fungal cells are fully eukaryotic, which means they contain a true nucleus surrounded by a nuclear membrane. Inside that nucleus, the DNA is wrapped around histone proteins just like in plants and animals. In addition to the nucleus, fungal cells contain mitochondria for energy production and a complex internal membrane system that includes the endoplasmic reticulum and Golgi apparatus. Unlike plants, fungi do not have chloroplasts and they do not carry out photosynthesis. Even so, many fungi are bright colored. These colors come from pigments located in the cell wall, not chlorophyll, and these pigments help protect fungi from damage caused by ultraviolet radiation in the environment. Fungal cells are surrounded by a thick and rigid cell wall, similar in function to the plant cell wall but very different in composition. Instead of cellulose, fungal cell walls are built from complex polysaccharides called chitin and glucans. These materials provide structural strength and help protect the cell from drying out from being eaten by other organisms. Beneath the cell wall is a plasma membrane, which resembles other eukaryotic membranes but uses ergosterol instead of cholesterol to maintain stability. Most fungi are non-motile, meaning they do not move on their own. Fungi are remarkably adaptable and can be found in an impressive range of habitats. They live in seawater, freshwater, soil, and even on human skin and mucous membranes. Some of the most primitive fungi, the citrids, are found primarily in aquatic environments. Other fungi, such as species that live in the deserts or sandy soils, are well adapted to dry conditions. There are also fungi in the parasites coral reef and survive on marine ecosystems. Even with this diversity, the majority of fungi are found in forest floors. In these environments, fungi act as primary decomposers, breaking down fallen leaves, wood, and other organic material. Fungi have a very different approach to nutrition compared to animals. In fungi, digestion happens before ingestion. They secrete enzymes called exoenzymes directly into the surroundings. These enzymes break down large complex molecules outside the organism into smaller, simpler molecules. Once digestion has occurred externally, the nutrients are then absorbed through the extensive surface area of the hyphae and the mycelium. This method allows fungi to efficiently extract nutrients from the materials like wood or leaf litter. Most fungi function as saprobes, meaning they obtain their nutrients from dead or decaying organic material, especially plant matter. This role is critical in ecosystems because fungi are among the few organisms capable of breaking down tough substances like cellulose and lignin found in wood and plant cell walls. They accomplish this by releasing powerful exoenzymes that convert those complex, insoluble compounds into smaller molecules such as glucose. As fungi absorb these nutrients, important elements like carbon and nitrogen are released back into the environment. This recycling process makes nutrients available to plants and other organisms and keep ecosystems functioning efficiently. Not all fungi are decomposers. Some species are parasites, meaning they obtain nutrients from living hosts. Parasitic fungi can infect plants, causing serious agricultural diseases such as smut and Dutch elm disease, which have had major economic and ecological impacts. Other fungi infect animals, including humans. Common example includes athlete's foot and yeast infections like candidiasis. In environments, these nutrients, such as nitrogen, are scarce. Certain fungi have evolved even more unusual strategies. They actively trap and digest small animals like nematodes, allowing them to survive in otherwise nutrient poor environments. Fungi are not always multicellular. Some species exist as single-celled organisms known as yeast. These unicellular fungi are especially important in medicine and industry. In addition, some fungi are dimorphic, meaning they can switch between unicellular and multicellular forms depending on environmental conditions, such as temperature and nutrient availability. This flexibility allows them to survive in very different environments and, in some cases, contribute to their ability to cause disease. A well-known example is Candida, which can exist harmlessly in the body but shifts from form under certain conditions and can cause infection. Most fungi exist as multicellular organisms and spend the majority of their lives in vegetative growth stages. During this stage, the fungus is composed of long, thin, thread-like structures called hyphae. These hyphae grow and branch extensively, forming a dense network known as mycelium. The mycelium is usually hidden within soil, wood, or other organic material and is responsible for nutrient absorption. When conditions are right, fungi enter the reproductive stage and produce structures that are more visible, such as mushrooms or spores producing bodies, which allows the fungus to reproduce and spread. In many fungi, hyphae are divided into individual cells by an internal cross-wall called septa. These septa are not completely solid. Instead, they contain small openings that allow nutrients, ions, and small molecules to move quickly from one cell to the next. This internal flow helps fungus function as a coordinated whole rather than an isolated cell. Some fungi, such as bread molds, do not have septa at all. Their hyphae are described as coencytic, meaning they form long tubes filled with many nuclei within a shared cytoplasm. Fungi generally thrive in environments that are moist and acidic, although they can be capable of growing in a wide range of conditions. Light is not required for fungal growth, which allows them to live underground or inside other organisms. Their metabolic needs vary when it comes to oxygen. Most fungi are obligate aerobes and require oxygen for survival. Some species, particularly certain citrids found in the digestive system of animals like cattle, are obligate anaerobes and cannot tolerate oxygen. Yeast falls in between these extremes. They are facultative anaerobes, growing best with oxygen but are able to thrive without it switching to fermentation, a process humans use to make beer and wine. It has flexible reproductive strategies that can reproduce either sexually, asexually, or both. Species that reproduce using both methods are referred to as perfect fungi, while those that reproduce asexually are called imperfect fungi. Regardless of the mode of reproduction, fungi rely heavily on spores. Those spores are lightweight and produced in very large numbers, allowing them to travel long distances through the air or by hitching rides on animals, producing so many spores that at least one will land on an environment with right conditions for growth and survival. Asexual reproduction is very common in fungi and can occur in several different ways. Some fungi reproduce when fragments of hyphae break off and grow in new individuals. Others produce asexual spores. Yeast, which are unicellular fungi, typically reproduce by a process called budding. During budding, a small bulge forms from the surface of the parent cell. The nucleus divides by mitosis. One copy moves into the bud. As the bud grows, it eventually separates from the parent cell, forming a new, genetically identical individual. And grow by mitosis to form multicellular haploid individuals. When sexual reproduction occurs, specialized cells or hyphae fuse, eventually leading to the fusion of nuclei and the formation of a diploid stage. This diploid phase is usually brief. Meiosis then occurs, producing new haploid spores that can be dispersed and begins the cycle again. Many fungi produce specialized reproductive structures that are designed to efficiently release spores into the environment. In some molds, spores are formed inside the structure called sporangium, which sits at the tip of the specialized hypha known as the sporangiophore. When the sporangium matures, it ruptures and releases spores, allowing them to disperse through air currents. This strategy is highly effective for spreading offspring over wide areas. Mold such as mucor, which is commonly found indoors, uses this type of. The citrids classified by phylum Citridiomycota are considered the most primitive group of fungi. It suggests they appeared more than 500 million years ago. Most citrids are unicellular, although few species form multicellular structures. Flagella act as members of the phylum Zygomycota, primitive saprobes that feed on decaying organic material such as dead plants. Species act as parasites. Their hypha are coencytic, meaning they lack septa and contain many haploid nuclei with a shared cytoplasm. During the vegetative stage, these nuclei remain haploid. Zygocetes commonly reproduce asexually by producing sporangiophores, which are released from the sporangia and quickly colonize nutrients. Interact and nuclei eventually fuse, briefly forming a diploid nucleus. Phylum Basidiomycota are the most recognizable, including mushrooms, puffballs, and bracket fungi. Their defining feature is the basidium, a club-shaped reproductive structure found at the tips of certain hyphae. Basidia produce haploid spores called basidiopores, which are meant for dispersal. These play a critical ecological role as filters, particularly in forestry. Basidies have a distinctive life cycle that includes prolonged dikaryotic stages where each cell contains two separate haploid nuclei. This stage can persist for a long time and makes up much of the fungal body. Eventually, nuclear fusion occurs within the basidium, forming a diploid nucleus. This is immediately followed by meiosis, which produces haploid basidiospores. Once released, these spores can germinate and grow into new haploid mycelia, continuing the life cycle. The extended dikaryotic phase is a key feature that sets Basidiomycetes from other fungal groups. Glomerulomycota is a relatively small phylum, but its ecological importance is enormous. Members of this group form arbuscular mycorrhizae, which are mutualistic associated with plants. Deuteromycota is not a true evolutionary group, but rather a polyphyletic category that historically includes fungi with no known sexual stage. These fungi reproduce asexually, most often by reproducing conidiospores. In some species, hypha form from different individuals confused, forming heterokaryotic hypha that contain genetically distinct nuclei. Advances in DNA analysis have shown that many deuteromites actually such as Ascomycota or Basidiomycota. As their sexual stages are discovered, these fungi are gradually reclassified into the appropriate evolutionary groups. Fungi are one of the most important decomposers in ecosystems. They specialize in breaking down dead organic matter, including materials that many other organisms cannot digest. As fungi grow, their hyphae penetrate in wood, soil, and plant membranes, secreting enzymes that dismantle complex molecules. This process releases essential nutrients such as carbon, nitrogen, and phosphorus back into the environment where they can be reused by plants and other organisms. The mushrooms or bracket fungus we see is only the reproductive structure. Most of the real work is happening out of sight in the extensive mycelial network. Humans have relied on fungi for thousands of years. Yeasts are essential for the production of beer and bread through fermentation. Fungi have also a critical source of medicine, most famous antibiotics like penicillin, which fungi produce to compete with bacteria. Beyond antibiotics, many other drugs have been isolated for fungal species. Fungi are also organisms for scientific research because they are relatively easy to grow and study. In addition, many fungi are consumed directly as food, including mushrooms that are nutrients and contribute to global food systems. Most plants on Earth form mycorrhizal relationships with fungi, making them one of the most widespread mutualisms in nature. The fungal mycelium extends far into the soil, greatly increasing the surface area available for absorbing water and minerals such as phosphorus. These nutrients are transported directly into the plant roots. In return, the plant supplies the fungus with carbohydrates producing during photosynthesis. This exchange benefits both partners and improves plant growth, stressing tolerance and survival, especially in nutrient poor soils. Another important form of fungus plant mutualism involves endophytes, which are fungi that live inside plant tissues without causing disease. These fungi can provide significant benefits to their host plants. Some endophytes produce chemical compounds that repel herbivores, while others increase resistance to pathogens, drought, or toxic metals in the soil. In return, the fungus gains access to nutrients and stable environments inside the plants. These hidden partnerships often go unnoticed, but they play a major role in plant health and ecosystem stability. Lichens are not single organisms, but stable partnerships between fungus and photosynthetic partners, either an alga or a cyanobacterium. The fungal components provide structure, protection, and moisture retention, while the photosynthetic partners produce sugars through photosynthesis. This collaboration allows lichens to survive in some of the harshest environments on Earth, including rare bare rocks, deserts, and polar regions. Because lichens are also sensitive to pollution and environmental change, they are also valuable indicators in ecosystem health. Lichens have a very specific internal structure that relates to fungus and photosynthetic organisms. The outermost layer, called the packed fungal hyphae, and functions as a protective barrier. Beneath that is the algozone, where photosynthesis takes place and sugars are produced. Below this layer is the medulla, which consists of loosely arranged fungal hyphae that help with gas exchange and water retention. The lower cortex provides a distribution and often contains rhizomes, which anchor the lichen securely to rocks, soils, and tree bark. Some of the most fascinating fungal mutualism occurs within animals. A classic example involves leaf-cutting ants in Central and South America. These ants harvest leaves not to eat directly, but to cultivate fungi in underground gardens. The fungi digest cellulose in the plant material, something the ants cannot do on their own. The ants then feed on the fungal growth. In return, the fungi receive a constant supply of fresh plant material and protection from competitors. Fungi engage in a wide range of symbiotic relationships with organisms. In parasitism, the fungi benefit at the expense of the host. When fungus cause disease, Many fungi that live or within organisms fall into this category. These interactions are especially important because parasitic and pathogenic fungi can agricultural and medical problems. Fungal infections in plants have had profound impacts on human history and food security. Many plant pathogens are fungi that infect tissues, disrupt nutrient transport, and eventually kill the host plant. These infections can destroy crops in the field or cause stored food to rot after harvest. Some fungi also produce powerful toxins that contaminate food supply, making them unused to consume. Large scale crop losses caused by fungal diseases can lead to economic hardship and even famine, highlighting the importance of understanding and managing fungal plants. Fungi can affect human health in very different ways. A mycosis refers to a fungal infection that damages tissues directly, such as skin or lung infections. Mycotoxicosis occurs when people consume foods contaminated with fungal toxins, while mousticemus results from eating poisonous mushrooms that contain toxins. In addition to infections and toxins, many people experience allergic reactions to fungal spores, which can trigger serious respiratory symptoms. While some fungal diseases are mild, others can be severe or even life threatening, especially in vulnerable populations. Fungal infections in humans can range from mild surface level conditions to serious internal diseases. A common example is ringworm, which appears as a red circular rash on the skin even though no worm is actually involved. Some fungi especially target hair and scalp tissue, such as Trichophantom violicium, which causes superficial infections on the scalp. Other fungi are inhaled and affect internal organs. Histoplasma is an example of the ascomycetes that infects the lungs and produces symptoms that can resemble the flu. Opportunistic fungal infections are caused by fungi that are normally harmless or already present in the environment or the human body. In healthy individuals, these fungi are kept in check by immune systems. However, in people with weakened immune defenses, such as those undergoing chemotherapy or organ transplantation, these organisms can lead to serious infections. Opportunistic fungi can invade tissues and spread throughout the body, making them difficult to treat. Because of this, they are a major concern in hospitals and other healthcare settings. Antifungal medications are essential for treating serious fungal infections, but an increasing number of antifungals are becoming resistant to other drugs. Antifungal resistance occurs when fungi survive and continue to grow despite treatments. This problem is especially concerning with invasive infections caused by Candida species. As resistance increases, treatment options become more limited, leading to longer illnesses or higher risk of complications. Antifungal resistance is now recognized as an emerging global health issue with parallels to antibiotic resistance in bacteria. Antifungal resistance can arise for several reasons. Some antifungal species are naturally resistant to certain medications, while others develop resistance through repeated or improper exposure to antifungal drugs. Using doses that are too low or stop treatment too early allows resistant fungi to survive and multiply. In addition, the use of antibacterial medications can disrupt normal microbial communities, creating conditions that favor fungal overgrowth. One of the most effective ways to slow resistance is through appropriate and responsible use of antifungals and antibacterial medications. Developing antifungal medications is often more difficult than developing antibiotic medications because fungal cells are eukaryotic, just like human cells. This means fungi share many cellular structures and biochemical pathways with us. As a result, it is harder to find drugs targets that harm the fungus rather than damaging the human cell. In contrast, bacteria are prokaryotic and have structures that are very different from ours, making them easier to target selectively. This fundamental biological similarity explains why antifungal drugs development is especially challenging.